We may have a somewhat abbreviated content just because we have a speaker missing in action, which is why we delayed a little bit. But we're going to go ahead and, in the interest of time, to get started. So it's my pleasure to talk now about TUBB4A-related leukodystrophy. We benefited from a multidisciplinary work group, including both our advocacy partners, our basic science group, a group focused on clinical outcome measures. That's part of a collaborative team that's working on a retrospective natural history study in TUBB4A. And also from some industry partners that are interested in developing therapeutics. And then finally also from members of a clinical advisory board that's working towards planning for clinical trials for the reasons that you'll see during our talk. TUBB4A leukodystrophy is a spectrum of disorders that, like many of the disorders we've been talking about the last 2 days, is really a lifespan disorder. And the MRI findings range from a very severe sort of early onset phenotype with severe hypomyelination plus or minus cerebellar atrophy through a childhood onset version with typically hypomyelination and atrophy of the basal ganglia and cerebellum, which is where the term HABC comes from, all the way to an adult onset dystonia with no visible imaging abnormalities whatsoever. So like many of the disorders we've been talking about, there's likely a range of cellular phenotypes and therefore clinical manifestations. It's inherited in a sporadic or autosomal dominant fashion depending on the severity. There's a single recurrent mutation that represents 20% of affected individuals in pediatric cohorts. That is a D249N variant, and that's closely associated with the phenotype of HABC, which was originally the pediatric leukodystrophy phenotype that was described. Over time, individuals progress, likely due to neuronal degeneration based on our animal models, and patients can become G-tube dependent and lose speech and ambulation. There's a strong component of dysarthria and dysphonia, so that some children, even who are still motorically fairly intact, and adults who are motorically fairly intact, will have a whispering dysphonia and have issues with the ability to produce a sound. There are no current clinical trials. And on a clinical basis, although advocacy and collaboration between our advocates and clinician scientists is a real strength of the community, and we have multiple registries and multinational natural history studies are ongoing, we still have a lot of work to do in understanding how that impacts clinical management. We also have a lot of good luck with preclinical models because they replicate the phenotype fairly well, which as we've seen in talks is not always the case. And so they reproduce both the phenotype of neuronal injury and hypomyelination. They reproduce the loss of cerebellar granule cells and striatal neuronal injury that is seen in very nice work done by Mahine Bougiani and others on the pathology of this disorder. However, we need to clarify more about how we get from microtubule dysfunction to those cellular phenotypes. And part of the reason why that isn't as clearly defined is really because those experiments are very difficult. Both biochemical and polymerization and sort of other assays related to microtubule function are technically challenging, and that expertise is still very much needed in this field. Another thing that's going to be very difficult from a research perspective is the significant phenotypic heterogeneity that I described earlier, because you're not going to be able to define an outcome measure that works for the early infantile phenotype and that also works for the adults. And so I think that thinking together across that pediatric and adult spectrum is going to be important as we think about clinical outcome measures that might be useful for clinical trials. Biomarkers for target engagement and therapeutic response are still lacking, and correlation of imaging features with clinical phenotypes is lacking because our most severe patients might have less phenotypic imaging involvement than those patients who present later on after a period of normal development. So from a clinical perspective, we're still probably lacking in diagnosis. Most patients are not diagnosed based on recognizable clinical phenotypes because when children present, they might not yet have the full imaging characteristics, and so they can present really with very nonspecific, delayed appearing myelination early on. And a lot of our patients are identified based on next-gen sequencing approaches, which, as we saw in talks earlier today, are not available worldwide and also are not available across the age spectrum. So it tends to be much more difficult for adults to get access to the type of genetic testing that is needed to accurately diagnose these individuals. Newborn screening will probably require molecular testing implementation because there's no clear way that you'd get a blood-based mechanism into a mass spec approach. And symptomatic management approaches are not well defined. Much as we sort of discussed with 4H, people are trying different things like deep brain stimulation, SDR, backflip pumps, and orthopedic surgeries to manage the very significant dystonic and extrapyramidal symptom complex seen in this disorder. And the impact and outcome of these interventions is not yet well understood. And then finally, the clinical phenotypes are not well stratified, limiting ability to provide prognostication for families at time of diagnosis, because within the group of patients, just as a small snippet of who present, with an initial period of early development, you have one group that we know retrospectively might progress very rapidly over the course of a couple of years to significant motor impairment, need for G-tube support, and things like that, and another group that might, maintain ambulation for a very long time. Multidisciplinary care teams will be needed to manage symptoms, and in particular, the advent of new clinical trials once they are possible. So when we look at our heat map and overview, we're grateful for our strengths, but we also recognize that therapeutic development is a significant gap right now, and that access to those therapies will be important, in particular as we think about the age spectrum. So we've picked, in our successes and challenges, we've picked two presentations. One which is focused on the preclinical models, their successes, and the explorations right now in therapeutic interventions. And then the second, which is a presentation on specifically the adult phenotype of this disorder, so that we sort of expand our understanding and thinking into the adult age spectrum. So it's now my pleasure to introduce Dr. Sunetra Sase from the Children's Hospital of Philadelphia, who's going to talk about her work in the therapeutics in preclinical models in HABC. Thank you very much.
Thank you, Adeline. I have been working with this model since last 4 to 5 years, and I'm really happy that we have come here, that we have a successful model now. And before beginning, I would like to give a nice introduction, but Adeline already explained that it is caused by the mutation in gene beta-tubulin 4A, and TUBB4A is exclusively or predominantly expressed in the brain, and 60% of total beta-tubulin comprises of TUBB4A. So the importance gets there, where you can see how much it is expressed in the brain, specifically in the myelinating oligodendrocytes and newly formed oligodendrocytes, with some extent in the OPCs, which are precursor cells and neurons. So this TUBB4A dimerizes with alpha-tubulin to form microtubules. And the microtubules are then important for neuronal polarity, axonal outgrowth, synapse formation, myelin formation, and for the oligodendrocyte functioning. So then mutations then cause a different spectrum of diseases. And one of the spectrum is HABC, which is hypomyelination with atrophy of basal ganglia and cerebellum. So this mutation, which is D249N, is a recurring mutation, and as Adeline mentioned, it is found in more than 20% of the patient population of TUBB4A-associated leukodystrophies, and it's suspected to cause toxic gain of function. It typically begins in toddler years, where patients have dystonia, ataxia, and the motor function is so progressive that kids lose the ambulation before the end of the first decade of life, since they are diagnosed. There is associated myelin loss and progressive striatal atrophy, which is shown here on the right side of the image, where the dotted line is hypomyelination and thin line is atrophy of basal ganglia. And if you can appreciate here, the cerebellar atrophy for the HABC. And there are no treatment approaches available. So to find the treatment, we need the models. And that's what, since last few years, we have been doing. And we have now mouse models, as well as we have the iPSC models, the cell models, which we are not presenting here just on the sense of time, but this beautiful model recapitulates HABC phenotype, which was published in eLife in 2020. So we generated this model in a heterozygous form, and heterozygous has mild phenotype, but here I'm showing you the homozygous mouse, which shows the phenotype starting from P9, and this is wild type, and this phenotype gets severe, where if you can see at P21, they are pretty tremulous and they walk with ataxic gait. The phenotype gets severe where they get highly dystonic, they don't ambulate that well, they have seizures, and they die from the seizures, where we have plotted the Kaplan-Meier survival curve, where they die around P30 to P35, and they also present with hypomyelination and cerebral atrophy, which I'm coming across. So to switch a little bit gears, which is not like completely switching, but to understand the function, we generated the knockout of this, or we didn't, but we ordered the knockout, and what we did is we double-crossed them to obtain the double knockout, which were completely viable, and they were breeding fine. So just to give you overview, now we characterize these four models along with our severe double mutant model. So we have the double knockout. This model has one copy of mutation and no TUBB4A. And we compared these all models to really see if we can rescue the phenotype or HABC phenotype. So this is survival in here, and if you see the knockout are completely normal, they live as long as wild type, they are happy, there is no any behavioral issues, we have done long-term behavioral issues, etc., etc. And then if you see the D249 knockout or the compound heterozygote, which I'm going to call, it has like 3 to 5 times better survival than the double homozygote. We did the behavior on them, which are like different series of tests, but this is like the tremor test, like I mentioned, and we just measured the tremor amplitude, and these are mainly to just set the outcome measures so that when we establish the treatment, we could use this. So we have like nice tremor measure where they show severe tremors, but then they are deceased. But these D249 knockout have much less severe early on, phenotype early, but then it gets there. Even the rotor, which is a motor dysfunction paradigm, so this double mutant have, are, as I said, they are less ambulatory and they are not performing well at this point, while the D249 and knockout can perform better early on, but they progress, or they have progressive motor dysfunction, but it's much more attenuated as compared to the double mutant. The second we checked was the hypomyelination, because that's what we see in patient population. And for the hypomyelination, we chose these different ages. Just to justify, the myelination in the mouse begins by P0, then it peaks at P21, and it completes around P48. And we chose these other time points. These specifically, this is end stage of this double mutant, and then this is end stage of the compound heterozygote. And we did myelin assessment using eriochrome cyanine stain, which stains the myelin, which is the blue in color here. And if you can see, there is much more profound loss in this double mutant versus there is preserved myelination and no loss in knockout, which is plotted here as a graph at different ages. We also did electron microscopy in optic nerve as well as in corpus callosum at two different end stages. And if you see, these are beautiful axons, and there is nice myelin around here, which is much more deteriorated in the double mutant. And we have plotted here as percent of myelinated axons, where there are thin myelinated, normal, and unmyelinated. And the unmyelinated axons were a lot more in the double mutant, while in this compound heterozygote, there was more thin myelinated axons. And of course, by the end stage, it looked like they had more unmyelinated, but still there was some preservation of thinly myelinated axons. So this again shows that how with the knockout we can rescue this phenotype. Then we also checked if we can, what we see at the oligodendrocytes. So we already know that the oligodendrocytes are reduced in the double mutant model, and we wanted to check if to what extent this compound heterozygote has loss of oligodendrocyte or reduced number of oligodendrocytes. And we do see that they are lost, or they are reduced, but they are well preserved as compared to this double mutant, and there is no oligodendrocyte phenotype in the knockout, which again is take home message is the knockout is really good model where we can see that it is a viable treatment strategy. Again, we checked what happens with the cerebellar atrophy. So we again checked these time points, and just to give overview, this is the mouse cerebellar architecture, which is stained by the neuron, and you can see a nice staining of the neuron, which has stained the granule neurons. And if you see, this is like the area for the double mutant where they have lost many cerebellar granule neurons, and the death occurs, which is like plotted here as the caspase, and we have also counted the neurons. And obviously by the end stage of this D249 knockout, we do see the loss of the cerebral granule neurons, but it's much more less severe than what we see in the double mutant. So this again, like same message, that it rescues the cerebellar atrophy, hypomyelination, and the motor phenotypes. So this shows that how we can use this model, how we can use this approach as an advantage to find the clinical treatment. So there is also this paper, this is a recent publication which resonates with our finding, where they have used another HABC mutation, which is N414K. There are very few patients for this mutation, and they show similar thing with the knockout or normal, and more the dosage of the mutation, more is the toxic gain of function or poisoning tubulin, which causes the defects and causes the phenotype to be severe, intermediate to severe. So this, like with the one, like just if I'm having one copy of wild type, the phenotype is mild, and that's what we see as well. And then again, this is JIT-null, which is N414K mutation, and then which is similar to our compound heterozygote, which has like in between phenotype, and this is the strongest phenotype. So as I said, this is a viable treatment approach, and that's why we focused on antisense oligonucleotides. And I think the group now already knows what are antisense oligonucleotides, where they just like bind to the target mRNA, recruit the RNase H, of course depending on the type of ASO you use, and then they are degraded and the mutated protein is reduced. And that's what we are aiming here, and we screen some ASOs against TUBB4A, and we have some pilot data which shows that we can see rescue in some of the animals. So we did the injections at P1 because we want to target the pediatric population, and the blue color here, 1316, is the ASO which we had screened, and all these other like black and gray are the controls. So this is just the video which shows at P35, basically they are like not ambulatory, and this mouse is still hanging. It's not perfect, it's not there yet, but it's still like better than the other mice which are there. And we can see here, like we have plotted this graph, and we see better rescue of the motor phenotype. We also did grip strength, and we saw better grip of these mice as compared to untreated controls. Then this is survival. We plotted the survival curve. The survival is not extensive, but we saw 7 to 8 days survival, better survival. And if you see these, this is I'm going to play with the video again. So this is much more ambulatory than the untreated, and it is exploring. It's behaving a little normal than the untreated mouse. So again, the amazing thing which we saw was the seizures, which we were not expecting at all. So these mice have really clonic-tonic seizures where they kind of like go stiff, and their tail goes like this, and when we counted the seizures over 12 hours, we see that there were no seizures or very few seizures in this mouse where we injected ASO. So that was like the amazing thing which we saw with this ASO. Then next we checked what is happening at the pathology level, and we do see some preservation of myelin both in corpus callosum as well as in cerebellum. And the data which I have not shown here is the cerebellar granule neurons, which we are not able to rescue, but we are currently synthesizing more ASOs and we are working on them. So we are hoping that we'll get there, but at least we see some proof of concept studies. So to conclude, this is a good model where we can use to develop the treatment strategy. Again, the knockout doesn't have any phenotype, which shows that these mutations are caused by toxic gain of function. Again, the disease correlates with the oral expression of mutant TUBB4A and relative preservation of wild type, and the knockdown is a viable treatment approach. As you see, that ASO treatment alleviates some of the HABC phenotypes. Again, treatment is not mutation specific, so it could be applicable to other TUBB4A associated leukodystrophies. And with this, I would really like to thank everybody, especially the lab team and our amazing research techs who have helped me to do this work. Louis is our IPS tech and Julia is our mouse tech who has done work, and then collaborators, and of course all the foundation partners, Synaptics Bio, who funds our research and affect patients and affected family. Thank you.
So thank you again, and thank you for inviting me. This is a very clinical talk, really. I probably should have gone before you, then we could have seen the patients and then your very nice mouse work, which really would be very good in the patients I'm about to show you. But it's to show you some of the phenotypes, and this is a very heterogeneous disorder. We see patients both with milder and severe phenotypes, as you've shown with the particular mutation in your mouse model. So I see all sorts of patients with neurogenetic conditions, and we video everybody. And this is why I like to show videos of patients as well. So as we've talked about, over 180 mutations in TUBB4. The main questions really are the genotype phenotype, can we predict progression, what scales perhaps to use, best management for patients, and talking about heterogeneity, which is a problem with many of these rare disorders. We don't quite understand why there's such a spectrum of clinical heterogeneity. On my last slide, I'll talk a little bit about newborn testing, which it may be slightly controversial and differs in different countries, but I think it's a very important thing to consider. So I'll run over this quite briefly, but basically clinically, MRI, but genetics. Genetics is changing the things we do. As we've spoke about, many people are testing older adults now genetically and picking up some unusual mutations and some unusual phenotypes. There's a differential here, including TUBB4. We've heard about some of the other genes today and yesterday. But TUBB4, we have two, what shall we say, a spectrum of phenotypes, from the predominantly DIP4, which was considered really a dystonia, but we consider it now probably more broader than that, to the HABC, which we've just heard about. I mean, this is not quite up to date in terms of mutations. There's many more mutations, but we have probably three sections of the gene. We have an N-terminus, second domain, middle part, and then a C-terminus. The N-terminus is where the first mutations were found, and I think that came out just about the same time as Adeline's paper in American Journal, was the finding of the DIP4, or the Heatmap family, which was very much a dominant dystonia, but quite complex. And as we've heard about with the TUBB4, the 249 mutation, which was first presented in the American Journal paper and the mouse model. So in terms of thinking about genotype, phenotype, I go back to a nice paper by Adeline and colleagues, and she also shared with me a nice poster looking at all of these mutations. And I think we can see a genotype-phenotype effect in terms of where the mutations lie, according to the age of onset, according to the imaging, and according to the clinical features. So if you have mutations in either end of the gene, the phenotype, in my opinion, is milder. The imaging tends to be less dramatic, as opposed to if you have mutations in the middle of the gene, which is the mutations of the 249 or the 255, around about that second domain. And so if we look at some of the clinical phenotypes, I'll put the volume up because we've heard about whispering dysphonia, but probably very few people in the audience know what whispering dysphonia is. So this is the original family, and they were first from a place called Heacham. I'll just stop the video. Heacham is just there. So I was born about 10 miles away from Heacham, and that's where the original family came from, and then they migrated over to Australia, probably as prisoners, and that's where the original family started. And most of the family now are in, I think, in a place called Townsville in Queensland. And this is the video of the patient.
The puppy bit the tape. We mow our lawn all year.
So this strained dysphonia, so it's vocal cord dystonia that's causing it, and it's very strained. It does sometimes improve with Botox in patients, not always, but it does.
You have to?
And this is patients with a mutation at the N-terminus, and it's the dysphonia, the dystonia, and then this classical hobby horse gait.
Excellent.
I've never ridden a hobby horse, so I couldn't tell you whether that was a hobby horse, but it's a very unusual gait, and it's quite classical in this particular mutation in this family.
And get in right close to the tongue if you can. The puppy bit the tape.
A puppy bit the tape.
Now this one. We eat eels every day.
So she improves with tetrabenazine, and some of the family also improved with DBS as well. And this seems to be a feature of patients at the N or the C terminus in terms of their response to treatment, as opposed to patients in the middle of the gene. So if I take you through to another patient, there's another patient with a mutation at codon 2. And on the left, preoperatively, this is why I quite like DBS in certain patients, and I think it improves anarthria in certain patients.
What did you say?
She's anarthric. And she's got an abnormal gait. And the main reason she went to DBS mainly was because of the anarthria. And the anarthria was improved a little bit, but the gait was significantly improved. And so was her dystonia rating scores. So that's the N-terminus. Now, if you look at the middle of the gene, this is where there's more of a severe phenotype. This is very much a spastic dystonia, early onset, ataxia, ocular abnormalities. And they often have this unusual, painful oculogyrate crisis, which we've seen in older patients. I couldn't tell you about the younger patients, but certainly it's present in older patients. And I'll give you just an example of this. Some of them do have the whispering dysphonia, but the features of patients with mutations in the middle of the gene, they tend to rapidly progress. This is a young girl from our series at Queen Square. Hopefully it will play. Maybe it won't. I'll describe what she sees. But basically, she's got a early-ish onset, childhood onset, not in the first year. I think onset was 2 or 3. She presents with a whispering, mild dysphonia, mild dystonia, and she does okay until she gets into her 20s, when she develops more of a generalized dystonia and spasticity. So it's and I think this kind of fits with some of Adeline's work in her poster, where they can be a little bit milder in the early phase, and then they progress more rapidly later on. And these are patients with mutations in the middle of the gene. And then if we look towards the C-terminus, again, very similar to the N-terminus patients. Earlier onset, slower progression, but often they get certain events that make them progress more rapidly, like this lady in her 50s.
And then can you lean your head like that? Good, and then the other side.
She's got this unusual crawling gait, and then when she uses a walker, it's kind of a hobby horse gait as well, similar to
rainbow is a...
And I'll just take you through that. So again, DBS helps her with her speech, helps her with her gait, and I think in terms of pre-DBS, this is quite dramatic for patients in terms of patients with an N or a C-terminal mutation. So thinking of the phenotypes, I think the main phenotype group that we have to consider without very much successful treatment are the patients in the second domain, in the middle of the gene, just like the mice that were presented. That's the area of need, and they would be the ones that I would want to target with ASOs. I mean, you should probably target any patient, but I think they're the ones that we have nothing for and don't respond to DBS, don't respond to your typical drugs such as your anticholinergics. They don't do at all well. And then Adeline also shared with me a poster, but only last night. So this is an addition, but she quite nicely looked at 180. I say she and team looked at 180 patients, including some of our patients from Francesca in London, and she found similar things that we found in terms of certain patients have an early infantile presentation, but then there's others, more commonly the late infantile presentation, where they don't do badly initially. I mean, this is sort of in the first few years of life, but they tend to rapidly progress later on in terms of their dystonia. They need a PEG feed, so there's obviously anarthria as well, swallowing problems, and that seems to be a limitation in the mutations in the middle of the gene. So we're all overall thinking about how to manage patients. I think, as we said, I think patients with certain mutations are very responsive to certain treatments, such as DBS. I think the dystonia, the swallowing, and the spasticity are the key things to look at in terms of improvement. I'm not sure about the imaging. There is some myelination abnormalities, but I'm not sure how susceptible that would be to improvement in terms of treatment, given the main features are atrophy, and I can't see ASOs actually putting that back into the brain of these patients. And then the other thing I want to mention is the TUBB4 phenotypic variation and the mosaicism. So many patients, or well, I say many, I know of 4, but that's only because of me thinking about them. I'm sure there's others. It's de novo, but when we use next generation sequencing, we see one of the parents has also got a percentage of mutations. So I've got 2 patients, one with 20%, another one with only 10%, but we see it on next generation sequencing. So is that part of the phenotype? Do we see that mosaicism, both gonadal and somatic, affecting the phenotype? Perhaps we're going to see parents presenting with late onset dystonia as well if they carry 20% of the mutation. We don't know this. And my last slide is just thinking about moving forward in our hospital and perhaps in your hospitals as well. So we have a system in our hospital where we take blood on everybody that consents, that walks in the door. In common disorders, we're looking at polygenic risk to predict problems, to prevent problems. So that's number 3 at the bottom here. Everybody walks in, gets that. I mean, they can say no, but they're offered that, and 99% of people take it. Then we have a second one where we take blood for Mendelian disorders, and we do whole genome on every patient that is part of the testing registry. So pretty much all inherited disorders, leukodystrophies, neuropathies, ataxias, spastic paraplegias. A part of that are genomes as a trio on the NHS, so they don't pay any money. But the thing we're doing at the moment over the next 3 years is doing a pilot of 500,000 newborns. And this is going to be a layer cake approach. So what we want to do is sequence these children. We will have actionable initial layer cake tests, such as treatable conditions, manageable conditions, and then later on we may consider talking about things like BRCA and Huntington's once they're over the age of 18. But under the age of 18 we will avoid any contentious issues such as Huntington's, BRCA mutations, and only deal with the actionable genes. So that's the plan with the genome sequencing. But then the other thing is talking about, some of you in the audience may have patients with a single allele in a recessive disorder. And what do you do if the patient fits that clinical picture? This is another initiative we have, is looking at RNA sequencing and then doing long-read sequencing. I've talked to a few of you about long-read sequencing in known genes as well as in that gene, because what we've found is deletions, inversions, insertions that actually, when you look at them, fit and can hopefully diagnose patients and then allow them to be part of a trial when they're genetically proven. I think the last slide is more contentious, but I think in 10 years we're going to be genome sequencing, certainly in the UK anyway, all babies, I imagine. It has to be careful in terms of how we report these mutations and how we look at them. There'll be problems. We'll probably get sued a few times, but I think it's very unlikely that this won't happen. Thank you very much.
Thank you. Questions? So, Kesar raised his hand first.
Really nice talks. Sunetra, I had a question for you. So in your obviously the double mutant mouse is more severe than the single mutation. And then when you say you cross the double mutant to the knockout, you say you kind of improve the phenotype?
Oh, sorry. We didn't cross double mutant. We crossed the heterozygous.
Sorry, the heterozygous.
Yeah.
So wouldn't the more appropriate comparator be the heterozygous mutation as comparing the heterozygous to the knockout?
Yes, so we do have that data, but I just didn't show. So we have like all the animal cohorts together, including the D249 and the wild type. And those are mild, those are at the milder side. That's why like I included the slide which included Carol's work,
Right.
which showed like similar what we see, which resonated with our work.
Right, because it sounds like the improvement in the phenotype when you cross it to the knockout is because you're reducing one mutant allele
Yes.
rather than knocking down the wild type allele.
Yes.
So I think the ASOs would be targeting the mutant allele rather than knocking down the total amount of tubulin.
Yes. ASOs is targeting like just all TUBB4A. It's just not targeting mutant TUBB4A.
Right.
Yeah.
What I'm trying to say is that the improvement is not necessarily because you're reducing the overall tubulin, but specifically because you're targeting the mutant allele.
But we have not tried that in D249 and knockout yet. So the ASO treatment which we have tried is in double mutant.
Right.
So when we try in the single mutant, but there will be no TUBB4A, so we will still decrease the mutant TUBB4A in the mouse.
But importantly, we importantly did not want to do personalized to one single mutation, even though we do have that common mutation, because we wanted the therapeutics to be accessible for all patients with TUBB4A mutations, right? So we are knocking down, it's very difficult because there's not great antibodies for TUBB4A compared to other tubulins because there's just a lot of homology. I won't get into the details, but we're not knocking down more specifically the mutant versus the wild type. We're just knocking down, and a knockdown is helpful.
Right.
And effectively what you're doing is you're permitting competition, and there's Drosophila data for this and things like that, but when you have any tubulin, not just TUBB4A, mutation, if you knock it down, it's well tolerated because other tubulins take its spot, right? So there's an upregulation of other tubulins, and then they're basically subbed in, right, for the missing tubulin, which is the mutant one or the wild type one of that type. Does that make sense?
Right. Yeah, I think maybe you can talk after this. But
Yeah.
I have another specific question.
And we also do see that data, like what Adeline is saying in our nanostring panel.
Yeah. So we see upregulation of other tubulins in response, and the TUBB4A wild type has has a phenotype, but it's much milder. And it takes so long that it's not really realistic from a preclinical model therapeutic perspective, because you'd have to wait a long time, right, to see effect. And so our primary goal in this is to develop a testing platform for therapies.
Also, TUBB4A, D249, and wild type has a mild, like just the myelin behavioral, but not the behavior phenotype, just the myelin degeneration.
Right. Okay. There's a question over here.
Is this working? I'm going to send it to Adeline because I know she can take my harder questions. But all of you can respond. When we're thinking about new therapies, moving beyond the gene and thinking more about what disease-modifying targeting. I didn't do anything. Okay. Remyelination, I know that's why I'm like not covering anything. Remyelination versus neurorepair. Which would you prioritize? Because each has its advantages and disadvantages, but I'd like to hear your perspective. We can't do everything, but it would be good to get that perspective on what would you target.
All right, I'll take a first pass, and then Henry, maybe you can also comment. So I think that, we've heard about a number of different hypomyelinating disorders this morning, right? And, in reality, it's likely that a lot of them have both a neuronal and a glial phenotype, right? And I think, and I love, Nicole, your example where you have the person with the worst cerebellar atrophy, but the better myelination is actually doing less well than the person with the better cerebellar atrophy, but the worse myelination, right? And so I think that we really need to think about probably and understand better what's driving the worst symptoms, right? And I think that it's not clear to me that the glial cells are driving the biggest symptomatic burden. And so I think we're going to have to think very carefully about cellular compartments as we develop these therapeutics. And I think, the adult data with the DYT4, where they don't actually have a visible, at least myelin phenotype, but have a very prominent neuronal phenotype, is a good example that you can be highly symptomatic. And, interestingly, we have patients with mutations at that same salt bridge R2, but who are early infantile onset. And they have some hypomyelination, but they don't have like the basal ganglia atrophy and everything else, but they have clinically a very prominent sort of extrapyramidal presentation, much like the adults. So I think there's a lot we don't understand about the cellular compartments, and I think that we're not going to tease that out until we start treating one compartment and see where our deficiencies are. I don't know if that is helpful, but I think that's the experience we're going to live through.
Yeah, I very much agree. I mean, there's a difference in adults and children, but yeah, I think it's interesting, though, you think of neuronal versus glial. Certainly in the adults, I'm not convinced that there's particularly any cognitive problems in these patients. I'm quite surprised that there's not. Correct me if I'm wrong from the audience, but so there is certainly a difference in that spectrum. But I agree with Adeline.
Yeah, and then within the neurons, there's different compartments of cortical, which is going to give you more dementia, and basal ganglia and cerebellar, which might give you these things are, which may be easier to treat. Yes.
I mean, I'm biased. I see mostly patients with that phenotype, so for me, they're easier, but not always.
Question, Florian.
Just curious, what are your mice dying of? And seeing that you're treating at P1, not seeing much extension of survival, is the cause of the death staying in the ASO treatment?
So the cause of the death, we believe, is the degeneration of the cerebral granule neurons, because we see the hypomyelination from the beginning, but the loss or death is mainly because of this degeneration of the granule neurons. And that's what we are trying now to target. Maybe if we could save them, then we would be able to reverse the phenotype much more efficiently than hypomyelination.
You think that
No, that's toxic gain of function. So it's starting somewhere from P14 to P21, and then it's getting worse by the end stage.
Yeah, conventional antisense approaches aren't equally penetrant across all cell types, right? And people know that as they've tried antisense approaches in different compartments, like Huntington's disease and things like that. And so I think that we're going to have to become more sophisticated in cell delivery as we sort out the best cellular targets.
Good question over there.
The antisense oligo, the treatment is very promising, so possibly can translate to the clinical use. I just wonder, just as Adeline said, is not every cell type is even penetrate. Is any area different of the penetration? How efficient the antisense oligo to equal the knockdown, right? So do you try different route or different dosage for compare the efficacy? And here's the question one. Second is for the mutation, the TUBB4A looks like they generate gain toxic function protein. So if you knock down, possibly have a threshold, not necessary to go completely eliminate protein if a threshold is there. Possibly you can test, how low the protein need maybe eliminate the phenotype.
Yeah, those are very good points. So to answer your first question, whether you're asking to use different routes and different methods, so we are looking into that, and we are currently exploring different route, which is cisterna magna, which is close to the cerebellum, to see if we can target the granule neurons. Then second is we are again trying different approaches like the AAV gene therapy, and also like other like nanoparticles like biomaterials to see if we can target the cerebral granule neurons. And then for the second thing is, yes, we do, but we don't see, like you say, we are not completely eliminating the protein. And unfortunately, we cannot see how much protein we have eliminated because we don't have very good antibody. But we are checking at the mRNA level, and in vivo, where we are seeing the knockdown is somewhere from 40 to 50%. And I think if we could like achieve that target everywhere in the brain, at least we would be able to have some intermediate phenotype, like the mouse model which I showed.
Thank you.
Do you want to add something?
Maybe last question.
So my question was, do you have any idea on the differential expression of the different cells in the central nervous system? Because different isotypes have different expression. If there is a bias in the expression of the different cell types in the central nervous system.
That's an excellent question, and I didn't show the data. So we have 4 different variants, and variant 3 is highly expressed, at least in humans. And in mouse, we are exploring that using RNA-scope, and have not shown that data. But we do see that the expression starts at P0 in mice, and it linearly increases like after P7, and then it kind of gets steady. Yeah.
And I think it is mapping, right? Again, these are experiments that are ongoing, but it's similar to the experiments that have been already done on single-cell RNA-seq, that it's highly expressed in cells of the glial lineage, right? But what we don't know is, it's maybe that a little bit of expression in certain cell types is sufficient to cause significant problems, especially at a specific developmental age.
Okay. All right. I think that's it. Thank you all.
All right. So just go ahead. I clap for my colleagues here.